Hi, good afternoon. My name is Meenakshi Saru. Today is 21st of August 2024, and I will be going through this very quick presentation about chemoreception, the sense of smell and taste. Okay. Once again, my name is Meenakshi Saru. 21st of August 2024, chemoreception, the sense of smell and taste. Now, first we're going to focus on olfaction, or the sense of smell. And you have a picture here of the diffuser and sense of smell. So we're first going to talk about the olfaction. And olfaction is the sense of smell. Now, within olfaction, you have a number of primary neurons. These primary olfactory sensory cells undergo something called bottom-up processing, in which they send signals to the brain for processing. Many of these primary olfactory cells converge onto a few secondary olfactory cells, as you can see here. But first, let's look at this first section here. And there are supporting cells. There's a basal lamina in which there is a high turnover of cells, and the highest turnover of cells actually occur within the olfactory section. And these olfactory sensory neurons, they converge into the olfactory bulb. And they are very small axons in these small sections because they are already located in the central nervous system. And in the olfactory bulb, you have the secondary dendrites and secondary cell bodies, which extend into the processing section of the brain. Now, as you can see in this section here, the olfactory neurons in the olfactory epithelium all are within and all deal with cranial nerve 1. And they all converge in the olfactory bulb, which passes through the olfactory tract and goes to the olfactory cortex. And we'll have more details about these convergence of the neurons in the next slide. Now, within the olfactory cortex, there can be connections to the cerebral cortex and further processing of the smells and the scents. Or you can have connections to the limbic system and talk about memory, talk about recall, connections of these scents and odor molecules. Okay, so this is a breakdown of the olfactory system and, well, a pictorial breakdown of the olfactory system. Now, in this slide here, we look at the main points. Now, odor molecules dissolve in the mucus and bind to the olfactory receptor proteins. Even though there is a limited range of odorants, there is a huge range of odor receptor proteins, and this can cater for the large range of odors that we smell on a daily basis and that we can recall. And these receptors are all G protein linked membrane receptors, and they can only pass on an action potential when the graded receptor potential passes, of course, a threshold. So enough stimulation to these membrane receptors would cause these graded receptors to pass the threshold, initiating action potential and sending it towards the secondary neurons within the olfactory bulb. Now, the signal, unlike other senses, do not pass through the thalamus and go straight to the olfactory cortex. So it doesn't have that initial processing that in between the intermediates within the whole processing. So because they're already in the central nervous system section. And the brain, even though it has such limited range of odorants or initial odor molecules that dissolve in this mucus and these receptor proteins, there is the combinations of these action potentials and the receptors, memory receptors that are triggered, can form many different categorizations of odors that are saved in memory for the limbic system recall. and also for our own feed-forward processes. And you know that the olfactory system, well, the olfaction is very much connected to gustation. So smell is connected to taste, and a lot of smells triggered a lot of further down behaviors such as um, salivating when you smell food, or feeling increasing your appetite, feeling the hunger, So a lot of these smells contribute to a lot of feed forward responses. And now we're going to talk about gustation, the sense of taste. And in gustation here, we have a lot of these receptor cells, which are also we have G protein coupled membrane receptors as well, just as we have in the smell. And a lot of these receptor cells are found bundled together within sections called taste buds along the surface of the tongue. Now, before it was further studied, there were people thought that different sections of the tongue had different focus on certain senses of taste. For example, more in the back section would have been for like sour or bitter or sweet. But as we learned today, that taste buds have all 4 or 5 senses within that section and are all stimulated at the same time by either the presence of sodium ions for salty, or presence of chloride ions, etc., etc. So we have the main senses of sweet, umami, bitter, and sour, right? And salty. And there's further investigation that needs to be done on which section of the taste buds and these bundles of G protein coupled receptor membrane proteins are specific to what, but there's a lot of research being done on this section of gustation. But the main thing is that once one of these G protein coupled membrane receptors bind to the necessary ions, they cause a signal cascade, signal transduction, which causes more ions to either flow into the cells or out of the cells, or movement of ions, which eventually triggers and eventually fires off primary sensory neurons and action potential to be sent to the brain. And we'll see that in the next slide. And in this slide here, we have a quick summary of gustation, or sense of taste. Now, just as in the smell, substances must dissolve within the mucus of the saliva, well, within the mouth, for it to trigger off these neurons. And these taste ligands, which are these substances that are dissolved, they have to interact with this membrane protein of the taste receptors. And there are 5 main ones, like as you saw before, clustered together. And whichever one is stimulated causes a signal cascade within that section of the cluster activating primary sensory neurons. Now, these gustatory neurons, these primary neurons, run through multiple cranial nerves, 7, 9, 10, to the medulla. And this sensory information passes through the thalamus to the gustatory cortex. And as we saw in smell, it doesn't pass through the thalamus, doesn't have that intermediate section. But in taste, it does. And the brain interprets these taste sensations of the most stimulated section of the cell responding neurons, causing the behavioral responses focus on that section of the cell that is most stimulated. And of course, you can see there's also feed-forward responses I used to talk about in the smell. So smell and taste are closely linked in this fact that they need each other. You can't truly taste food without first smelling it and having its odor molecules mixing. So those two senses are very much connected, and they're both found very close to the central nervous system. And such their secondary and the primary neurons have very short axons, and they go through very high turnover rates, and it is very easy to have a lot of memory stored based on gustation and smell. And you see there's a summary here. So olfactory pathway is, we'll look at this here. We have the olfactory epithelium, we have the cranial nerve 1, goes to the secondary sensory neuron in the olfactory bulb, you have the olfactory tract, and the cortex. You see there's no thalamus. And then you have the taste pathways, and you have these ligands activating these taste cells. You have many intracellular pathways, the influx of calcium ions, of sodium ions, depending on which section of the cell is most stimulated. And you have these primary sensory neurons signaling to the brain, to the medulla, the gustatory cortex. And you have the idea of population coding, in which the most, even though there could be an overflux of signals, sensory electrical signals passing to the brain, the ones that are most stimulated and most grouped together, population coding, are the ones that we will interpret the most. So if you know a food might have salty flavor, or very bitter, or very sweet taste, the one that is most strongly stimulated. And you have smell and taste. And the smell, the olfactory epithelium, is the basic epithelium, and the taste, you have the taste buds, right? And you have the supporting cells and everything. So in taste, you have the 4 cell types together in a taste bud, and in smell, you just have the olfactory sensory neurons, very short, high turnover rates, right? Then each taste bud can be used for all tastes. So everything is in one taste bud: the sweet, the umami, the bitter, the sour. But in smell, the olfactory receptors is very, very limited to the range of odorants, as we saw before. And in smell, you have cranial nerve 1, bypasses the thalamus, and in taste, you have cranial nerve, those 3 cranial nerves, right? And I would like to thank you for listening to my quick presentation on smell and taste and similarities and the differences. And I would like you to have a really good day. Thank you.